Right, I'm going to take epistasis in 3 parts, really. I think it makes more sense just to break it all up. It is tricky to get your head around. I think it's easier if we take one thing at a time. Now, I think the difficulty comes because you're probably comfortable with doing genetic crosses on this kind of idea where we've got a heterozygote organism. We've got 2 alleles on the go here. Sorry, 2 genes with the different versions of the alleles. And this is the kind of thing where we'd get the classic 9:3:3:1 ratio that we kind of expect to see, if you like. Now, this is looking at alleles that are responsible for a phenotype. So, you know, it could be that Y is, it could be pea plants, you know, Y gene is responsible, the Y allele is responsible for the height, and the Z is responsible for the colour of flowers, okay? You know, we get these things like tall and pink and short and red and short and pink and tall, or whatever it may be. Now, the problem with epistasis is we tend to get into that mindset. And in fact, most genes in an organism don't actually express a phenotype. What they do is they control the expression of another gene. And this is kind of what epistasis is talking about, that the presence of one gene can affect if another gene is expressed or not. So if we take an example of, and we'll make something up here, we'll have blue represented by a dominant allele, and the small allele is going to be white. So the phenotype here could be, I don't know, colour of feathers, for example. So what possible combinations would give us blue and what would give us white? Well, homozygous dominant would give us blue. Homozygous recessive would give us blue as well, because we've got the dominant one. Sorry, heterozygous. And homozygous recessive would give us white. And very often on these questions, they will be things about colour. And very often you end up with the recessive version, or if the gene isn't expressed, it becomes white. You know, perhaps this is actually a protein that's made. If you don't make it, the default colour, if you like, is white. The default is to have no colour at all. So it's very often about colour. So that's our allele for colour. This is sometimes, or is referred to as the hypostatic alleles. Hypo meaning under, okay? They are under the influence or under the control, in this case, of a dominant pair of alleles. So I'm going to put those ones in now, and I'll use a different colour. I think it's easier to spot this if I do it like this. So A is the dominant one and little a is the recessive one. If you have any dominant alleles present in the epistatic alleles, it masks the effect of what comes next. So in other words, as long as there's one present, it doesn't matter what comes next, it's going to hide it completely. And we'll put A allows expression. So the combinations here are homozygous dominant would mask, heterozygous would mask, and homozygous recessive would allow this gene to be expressed. So it doesn't matter what comes next. If the dominant allele is present, nothing will happen. You know, it will express as white. In other words, it will show up as white, the phenotype. Sometimes it's represented by just doing a dash. So A dash could be AA or AA. What that's saying is it doesn't matter what comes here. It could be anything, because as soon as you've got the dominant one, that's the one that counts. Okay. So if we now try to do this as like a Punnett square, we can maybe follow what's happening. And I'd recommend you do this if you're working your way through in a question. I always recommend on genetics questions writing the full thing out. So if we're going to do a cross, I'll keep with the colours here of AA, BB. There we go. So what possible zygotes, what possible gametes, sorry, could we get? And this should look like a classic dihybrid cross. We can have big A, big B, big A, little b, little a, big B, little a, little b, like this. This should look fairly familiar to you as a classic kind of Punnett square. Now I could, and I'd actually recommend this, I could work through every possible one of these combinations and then see what happens. And I would recommend you do that, you know, so I'd go through, that's going to be AA, that's going to be AA, that one's going to be BB, that's going to be BB. Okay, now I could go through all those. A tip for you, if you take to the exam a couple of different coloured pens, well, you can take a black and a pencil though, because it sometimes helps in these Punnett squares to mark off squares, you know, for example in a pencil, to maybe write in here what the phenotype is as you go along. And I'm saying that, I'm just looking for a pencil, which I don't have one. I was going to show you what I mean, never mind. I could write in here, you know, that one, okay, that is going to stop those expressing, so I know that the phenotype is just going to be white. Or I could just put W, same here, okay? Now I could work through all of those, or I could just look at them first of all and say, well look, that one's going to have, all of those are going to have a something in there, so all of those are going to be white, all of those are going to have a something in there, so all of those are going to be white. Doesn't matter what comes next, because as soon as I've got one of those dominant ones, nothing else happens, and all of that column, so I don't need to really do all of those out, I can see what's going to happen. Oh, and that one as well. Yeah, so you can see where I've got those. All I'm really interested in now is this box at the end. So A. What do I get here? Big B, big B, big B, little b, big B, little b, little b, little b. Okay. What's the phenotype here? Well, now I can refer back to this and I can say, well, blue, 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 and white. Let's use a different colour. Blue, 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 and white. Okay. Now, if in this case this completely stopped it from expressing, I would now have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 whites plus that white, which gives me a 13:3 ratio of white to blue. Okay. Depending on what you do, sometimes you will also get a 12:1 ratio, particularly in cases where white isn't going enough. This was something else, like if little b gave you, I don't know, for some reason it gave you pink, just for example, that would now be pink, so I'd have 12 white because nothing got expressed, 3 blue and 1 pink, so I'd get the, sorry, my ratio's wrong. 12 to 3 to 1, 33. Notice it always adds up to 16 because there are 16 boxes. So that's dominant epistasis. If the epistatic pair are dominant, if there's just one dominant, then it doesn't matter what comes after it, it's going to mask that effect.